હેલો ફ્રેન્ડ્સ, હું બારીયા દર્શન. આપણે આ પહેલાં 6 પાર્ટ પૂરા કર્યા. એમાં પહેલા પાર્ટમાં આપણે ઇન્ટ્રોડક્શન ડાયજેસ્ટિવ સિસ્ટમનું ને પ્રોસેસ ઓફ ડાયજેસ્ટિવ સિસ્ટમ જોયું. પાર્ટ 2માં જે એલિમેન્ટરી ટ્રેક્સ એના 4 લેયર જોયા. પાર્ટ 3માં ફેરિંગ્સ ને ઇસોફેગસ જોયું. પાર્ટ 4માં સ્ટમક અને પાર્ટ 6માં અને વચમાં એક રહી ગયું એમાં આપણે માઉથ જોયું. ટીથ અને ટંગનો સમાવેશ થાય. સ્મોલ ઇન્ટેસ્ટાઇન જોયું. આજે આપણે કેમિકલ ડાયજેશન થાય છે જે સ્મોલ ઇન્ટેસ્ટાઇનમાં એ જોવાનું છે. વેન એસિડિક કાઇમ passes into the small intestine, it is mixed with the pancreatic juice, bile and intestinal juice, and it is contact with the enterocells of the villi. In the small intestine, the digestive of the all nutrients is completed. Carbohydrates are broken down to monosaccharides, proteins are broken down to amino acids, fats are broken down to fatty acids and glycerol. Pancreatic juice. The pancreatic juice enter the duodenum at the hepatopancreatic ampulla and consist of water, mineral salts, enzymes, enzyme amylase and lipase, inactive enzyme trypsinogens, chymotrypsinogens and procarboxypeptide. Pancreatic juice is alkaline pH 8 because it contains significant quantities of bicarbonate ions which are alkaline in solution. When acid stomach contents enter the duodenum, they are mixed with pancreatic juice and bile, and pH is raised to between 6 and 8. This is the pH 8 which the pancreatic enzymes amylase and lipase act most effectively. Functions. Digestive of protein. Digestion of protein થાય છે એ પહેલું ફંક્શન છે. Trypsinogen and chymotrypsinogen are inactive enzyme precursors activated by enterokinase. બીજું નામ કહીએ તો enteropeptidase, an enzyme in the microvilli which convert them into the active proteolytic enzymes trypsin and chymotrypsin. These enzymes convert polypeptide to tripeptide, dipeptides and amino acids. It is important that they are produced as inactive processors and are activated only upon arrival in the duodenum. Otherwise, they would digest the pancreas. Next, digestion of carbohydrates. Pancreatic amylase converts all digestive polysaccharides, starch, not acted upon by salivary amylase to disaccharides. Digestion of fats. Lipids convert fats to fatty acids and glycerol. To add the action of lipase, bile salts, emulsified fats, for example, reduce the size of the globules, increasing their surface area. Control of secretion. The secretion of pancreatic juice is stimulated by secretion and CCK produced by endocrine cells in the wall of the duodenum. The presence in the duodenum of acid material from the stomach stimulates the production of these hormones. Next, આપણે bile જોવાનું છે. Bile secreted by the liver is unable to enter the duodenum when the hepatopancreatic sphincter is closed. Therefore, it passes from the hepatic duct along the cystic duct to the gallbladder where it is stored. Bile has a pH of 8 and between 500 and 1000 ml are secreted daily. It consists of water, mineral salts, mucus, bile salts, bile pigments, mainly bilirubin, cholesterol. Function of the bile. The bile salts sodium taurocholate and sodium glycocholate emulsify fats in the small intestine. The bile pigment bilirubin is a waste product of the breakdown of the erythrocytes and is excreted in the bile rather than in the urine because of its low solubility in water. Bilirubin is altered by microbes in the large intestine. Some of the resultant urobilinogen, which is highly water soluble, is reabsorbed and then excreted in the urine, but most is converted to stercobilin and excreted in the feces. Fatty acids are insoluble in water, which makes them very difficult to absorb through the intestinal wall. Bile salts make fatty acid soluble, enabling both these and fat soluble vitamins, for example vitamin K, to be readily absorbed. Stercobilin colors and deodorize the feces released from the gallbladder. When a meal has been eaten, the hormone CCK is secreted by the duodenum during the intestinal phase of secretion of gastric juice. This stimulates concentration of the gallbladder and relaxation of the hepatopancreatic sphincter, enabling the bile and pancreatic juice to pass into the duodenum together. A more marked activity is noted if a chyme entering the duodenum contains a high proportion of fat. Intestinal secretion. The principal constituents of intestinal secretions are water, mucus, mineral salts, enzyme, enterokinase. Most of the digestive enzymes in the small intestine are contained in the enterocytes of the wall of the villi. Digestion of carbohydrates, protein, and fat is completed by direct contact between these nutrients and the microvilli and within the enterocytes. The enzymes involved in completing the chemical digestion of food in the enterocytes of the villi are peptidase, lipase, sucrase, maltase, and lactase. Chemical digestion with associated with enterocytes. Alkaline intestinal juice pH 7.8 to 8, acids in rising the pH of the intestinal contents to between 6.5 and 7.5. Enterokinase activates pancreatic peptidase such as trypsin, which converts some polypeptides to amino acids and some to smaller peptidases. The final stage of breakdown to amino acid of all peptides occurs inside the enterocytes. Lipids complete the digestion of emulsification fats to fatty acid and glycerol, partially in the intestine and partly in the enterocytes. Sucrase, maltase, and lactase complete the digestion of carbohydrates by converting disaccharides such as sucrase, maltase, and lactose to monosaccharides inside the enterocytes. Control of secretion. Mechanical stimulation of the intestinal glands by chyme is delivered to the main stimulus for the secretion of intestinal juice. Although the hormone secretion may be also is involved. Next, absorption of nutrients. Absorption of nutrients occurs by two possible processes. બે પ્રોસેસ છે, ડિફ્યુઝન ને એક્ટિવ ટ્રાન્સપોર્ટ. ડિફ્યુઝન એટલે પ્રસરણ. દાખલા તરીકે આપણે રૂમના એક ખૂણામાં અગરબત્તી સળગાવી હોય તો બીજા ખૂણામાં જે સુગંધ અગરબત્તીની આવે એને ડિફ્યુઝન કહેવાય, પ્રસરણ કહેવાય. મોનોસેકેરાઇડ્સ, એમિનો એસિડ્સ, ફેટી એસિડ્સ એન્ડ ગ્લિસરોલ ડિફ્યુઝ slowly down their concentration gradients into the enterocytes from the intestinal lumen. બીજું છે એક્ટિવ ટ્રાન્સપોર્ટ. એક્ટિવ ટ્રાન્સપોર્ટ એટલે લોઅર કોન્સન્ટ્રેશન ટુ હાયર કોન્સન્ટ્રેશન જવા માટે જે એનર્જીની જરૂર પડે એને એક્ટિવ ટ્રાન્સપોર્ટ કહેવાય. મોનોસેકેરાઇડ્સ, એમિનો એસિડ્સ, ફેટી એસિડ્સ એન્ડ ગ્લિસરોલ may be actively transported into the villi. This is faster than diffusion. Disaccharides, dipeptides, and tripeptides are also actively transported into the enterocytes, where their digestion is completed before transfer into the capillaries of the villi. Monosaccharides and amino acids pass into the capillaries in the villi, and fatty acids and glycerol into the lactyle. Some proteins are absorbed unchanged. For example, antibodies present in the breast milk and oral vaccines such as polymyelitis vaccine. The extent of protein absorption is believed to be limited. Other nutrients such as vitamins, mineral salts, and water are also absorbed from the small intestine into the blood capillaries. Fat soluble vitamins are absorbed into the uh, lactales along with fatty acids and glycerols. Vitamin B12 combines with intrinsic factor in the stomach and is actively absorbed in the terminal ileum. The surface area through which absorption takes place in the small intestine is greatly increased by the circular fold of mucous membrane and by the very large number of villi and microvilli present. It has been calculated that the surface area of the small intestine is about 5 times that of the whole body. હવે આપણે ડાયાગ્રામ જોઈએ. The absorption of nutrients. Lumen of intestinal carbohydrates, polypeptides, fat molecules, goblet cells, blood capillaries છે, central lacteal છે, intestinal gland, blood in capillaries, lymph from lacteal containing fat. Large amount of fluid enter the alimentary tract each day. Of these, only about 500 ml is not absorbed by the small intestine and pass it into the large intestine. Next diagram. Average volumes of fluid ingested, secreted, absorbed, and eliminated from the gastrointestinal tract daily. Water 1200 ml, saliva 1500 ml, gastric juice 2000 ml, bile 500 ml, pancreatic juice 1500 ml, intestinal juice 1500 ml, કોલનમાં 1400 ml અને ફીસીસ છે એ 100 ml સ્વરૂપે નીકળી જાય છે. એમાં એબ્ઝોર્પ્શન 6700 ml અને 1400 ml 6000 એ એબ્ઝોર્બ થાય છે બ્લડ સ્ટ્રીમમાં અને ઇન્ટેસ્ટાઇનલ જ્યુસમાં તે 1500 ml